Good morning, everybody. It's my great pleasure today to introduce our guest speaker, Dr. Linus Pauling. As everyone knows, Dr. Linus Pauling is one of the greatest scientists of all time. He has made very important discoveries in nuclear physics, biology, immunology, and nutritional treatment of cancer and other diseases. He's written more than 1,000 papers and books. He has received presidential medals from President Truman and President Ford. He's received more Nobel Prizes than any other person. He's received honorary doctorates from 48 universities. He's served as a professor for many fine institutions, including here at Stanford, and he's now the research professor at the Linus Pauling Institute in Palo Alto. Today he's going to talk about vitamin C and heart disease, and we're all glad to have you. Thank you for coming to speak with us today, Dr. Pauling.
I think this is only the second talk that I've given about vitamin C and heart disease. I've given many talks about vitamin C in relation to health over the last 25 years, but only recently has my work extended significantly into heart disease. I'll start out by mentioning how it happened that I became interested in vitamin C. Not telling the whole story, but part of it. I gave a talk in New York, 1965, and in the course of my talk I said I'd like to live 25 years longer to be able to enjoy reading about the discoveries about the nature of the universe that scientists would be making during the next 25 years, just as I've been enjoying learning about similar discoveries for quite a while then. When I got back to California, I received a letter from biochemist Irwin Stone, in which he said he was sending me his reprints about hypoascorbemia, a genetic disease from which essentially all human beings suffer. And that if I followed his recommendations of taking 50 times the RDA of vitamin C, 3,000 milligrams a day instead of just 60 milligrams a day, I'd perhaps live not just 25 years longer, but 50 years longer. So I read these papers and was quite impressed by information and arguments that I just hadn't known before. So I began following that regimen of taking 3 grams of vitamin C a day, and observed, as did my wife too, that neither I nor she ever caught colds in the next year, say. And then I'd suffered from colds very much my whole life. So then I was asked to speak at the opening ceremony of a new medical school in New York, Mount Sinai. Give a 10-minute speech. 2 or 3 others, Beadle and a couple of others, were also giving short talks at this ceremony. I thought I must say something medical. What do I know? Well, that taking high-dose vitamin C helps control the common cold. So I made that statement. I got a very vituperous letter from a professor of medicine criticizing me and saying, can you point out a single randomized double-blind study that shows that vitamin C has any more value than a placebo? I finally got around to searching through the literature. I wrote to him saying I had found 4 studies of this sort. He just refused to pay any attention to these studies, and while I was reading through, I noticed that although one of the studies was mentioned in some of the textbook and books and reference books, the study was misrepresented. It was said that high doses of vitamin C have no value against a common cold, whereas in fact the investigators had found that their high doses, only 200 milligrams a day, cut down the amount of illness with the common cold by a third, which I thought was a significant effect worth following up. Well, by this time I'd got so interested in the history of the vitamins that I decided, and so irritated with this fellow, Victor Herbert, his unwillingness to look at the facts and discuss the matter, that I sat down and wrote my book, Vitamin C and Common Cold. But I'd also found in the literature reports that high doses of vitamin C were valuable for cancer patients as an adjunct to appropriate conventional therapy or inappropriate conventional therapy, either in various ways, improving the quality of life for the cancer patients, also cutting down on the side effects of the anti-cancer drugs. And so it happened that I got in contact with a physician in Scotland, Dr. Cameron, who had already started giving vitamin C to patients with advanced cancer. So when I retired as professor of chemistry at Stanford, we set up the institute where I work now for the purpose of studying vitamin C and other orthomolecular substances in relation to health and disease. And this work is still going on at the institute over on 440 South, 440, what is the address? Page Mill Road. We were on Sand Hill Road for a while and then moved to Page Mill Road. The aspect of vitamin C that impressed me the most, as discussed in the papers that Irwin Stone sent me in 1965, was something that I hadn't known. Plants in general manufacture all of the vitamins. Animals have to get these vitamins exogenously. Vitamin A, vitamin E, the various B vitamins. They rely upon the amounts that they get from eating plants. And the explanation presumably is that when the first animals began running around eating the plants, they had the ability to manufacture these important substances, but they really no longer needed this ability because they were getting them in their plant food, and mutants who had shuffled off the burden of the operating apparatus for synthesizing these substances had an evolutionary advantage over those who were burdened with this unnecessary apparatus. So that's why all animals require these vitamins. Well, the odd thing is that this isn't true of vitamin C. Perhaps 99 9/10% of all animal species continue to manufacture ascorbate. They didn't give it up. Why not? Presumably because to be in good health, they need to have more ascorbate than they get in their plants that they ingest, and animal foods too. So they continue to manufacture ascorbate. Well, we can ask, why do animals need more ascorbate than plants? with the other vitamins, this doesn't seem to be true, that animals need more than plants do. The answer is that animals use ascorbate in a way that plants do not, an important way for the animals, namely to synthesize collagen. Collagen is the macromolecule that holds animal bodies together. Plants don't need collagen because they use a polysaccharide, cellulose, a carbohydrate, as their principal structural protein. Animals convert procollagen to collagen by hydroxylating lysyl and prolyl groups in the procollagen molecule to hydroxylysyl, hydroxyprolyl. And in the course of this, for every hydroxylation, every hydroxyl group introduced, one ascorbate is destroyed. People who die of scurvy, half of them die of hemorrhages, internal bleeding, because of inability to synthesize the collagen that serves to strengthen the blood vessels enough to prevent their rupture. The other half die of infectious diseases which they are not able to withstand because of the importance of ascorbate to the body's natural protective mechanisms, the immune mechanisms and others. So this is an indication that human beings are unfortunate because of the inability to synthesize ascorbate, and as Linus STANFORD said, we all suffer from this genetic disease, hypoascorbemia, and are in poor health rather than what ought to be good health that would be associated with having the right amount of ascorbate in our bodies. Linus Stone, for 40 years, had followed the ascorbate matter, and he finally published in 1972 a book, The Healing Factor Vitamin C Against Disease, in which he discussed 100 diseases for which there's reported evidence in the literature that a high intake of vitamin C has some value in helping to combat the disease. Vitamin C is, of course, a specific preventive for scurvy, but perhaps not for any, well, there may be some other diseases, genetic diseases, say, that depend upon hydroxylation reactions or other reactions involving vitamin C, for which vitamin C could be said to be a specific prophylactic or therapeutic agent. But in general, the value of high doses of vitamin C in providing protection against disease is the result of the involvement of ascorbate in a great number of important biochemical reactions in the human body. Among these, we have the detoxification of toxic substances, where vitamin C, aided by enzymes in the liver, presumably, hydroxylates or glycosylates a toxic substance, and then it is eliminated in the urine. So the next point that interested me very much was how much vitamin C do these animals manufacture? Human beings, presumably a precursor of human beings and the other primates, had lost the ability to manufacture vitamin C because this precursor was living in a tropical valley on fruits and vegetables that had a very high content of ascorbate. You can get as much as 10 grams a day on a diet of just the high ascorbate fruits, a diet that provides 2,500 kilocalories of food energy per day. So then, of course, human beings moved into moderate and subarctic regions and began, where the food did not contain so much ascorbate, began suffering from hypoascorbemia and began trying to develop, through the process of evolution, following up random mutations that were favorable, ways to keep from dying of scurvy. And of course, human beings began thinking about the matter without knowing what it was that was responsible, and developed ways by making sauerkraut and pickles and so on of getting through the winter without having died of scurvy in the meantime. So other investigators have checked the amounts of ascorbate that animals manufacture. The amounts vary if they're proportional to body weight for different, if you calculate over to the weight of a man, 70 kilograms, the amounts range from around 2 or 3 grams a day per 70 kilogram body weight to 18 or 20 grams a day. That is between around 50 times RDA for humans and 300 times RDA for humans, the average being something like 10 or 12 grams per day. Why do animals, generation after generation, keep manufacturing this large amount of ascorbate? Presumably because it puts the animals in better health than if they manufactured smaller amounts. Well, we are so similar to other animals in our biochemistry that I can't believe that this high requirement for vitamin C applies only to these thousands of species of animals and not to human beings. So I think that the optimum intake of vitamin C is perhaps around 10 or 12 grams a day for a human being, and that if we want to be in good health rather than in ordinary poor health, it would be good to be taking this high-dose vitamin C. What happened next was about 1967, after I received letters from Irving Stone, I happened to run across papers by 2 psychiatrists in Saskatoon, in Saskatchewan. They were out in the country, I've forgotten where, at a psychiatric hospital. They reported that they could control schizophrenia to some extent by giving patients vitamins, especially vitamin B3, niacin, nicotinic acid or nicotinamide, and vitamin C. Well, I'd been working on schizophrenia and other mental diseases for 10 years. My project had come to an end. I didn't have a laboratory. I'd left the California Institute of Technology by this time. But I had some interest in mental disease. So, although I wasn't very much impressed at first, not being very interested in drugs and perhaps thinking of these vitamins as drugs, after a while, a week or two, I realized something was bothering me. I knew that drugs often have value against particular diseases, and that the more of the drug you take, the greater the chance is that it will be effective against the disease. But drugs are also toxic, can be lethal. So physicians prescribe drugs up at an amount somewhat smaller than the lethal dose, with the hope, if it's a serious disease, that the disease will be controlled. And here, what was bothering me was an interesting fact. Vitamin C and vitamin B3 have powerful physiological activity. A little pinch every day, a few milligrams every day of each of these substances will keep a person from dying of scurvy or pellagra, just as a little pinch of thiamine will keep a person from dying of very, very, very powerful substances in their physiological effects. And yet, Hoffer and Osmond in Canada, I realized, were giving patients as much as 1,000 times this amount that is physiologically active, that keeps people from dying. This is quite different from drugs, where the therapeutic dose is close to the lethal dose. So I asked myself a question. I was excited about this. I said, here I am, 66 years old in 1967. Here I am with interest in nearly everything that goes on in the world. And I've never heard about substances of this sort before that have a very wide range of tolerance by human beings and that have important physiological properties even when they are taken in very small doses. So there's a question in my mind, somewhere along perhaps is the amount of each of these substances that would put a person in the best of health. Not the amount that keeps people barely alive the way most people in the world are now, but the amount that puts them in the best of health. The optimum intake. And what is the optimum intake? Well, surely nutritionists and physicians have investigated this matter in the past. I couldn't find anything significant in the literature discussing the question of the optimum intake, except Irwin Stone, of course, who said the optimum intake was perhaps 10 or 12 grams of vitamin C a day, and he was being conservative, recommending 3 grams a day to people. So I thought, this is an interesting question. It's not so interesting as a lot of other scientific questions that I have worked on and get involved in, but nevertheless, it's an interesting question. What are the optimum intakes of these powerful substances? And moreover, much of the work that I do doesn't have any relationship to human health or the amount of suffering in the world, but if we were to work on this problem, we might well be able to make some discoveries that would have a really significant effect in cutting down the amount of suffering that people go through during their passage through their existence, their lives on Earth. So I thought we ought to be looking into this matter, find the optimum intake. I also thought there ought to be a name for these substances that are so different from ordinary pharmaceutical substances because of their lack of toxicity, substances that are normally present in the human body. I'll invent a name for them. I'll call them orthomolecular substances. Ortho meaning right, the right molecules in the right amounts. And the right molecules are the molecules that are normally present in the human body and required for life. For example, cholesterol is an orthomolecular substance. The health of essentially every cell in the body depends upon having the right amount of cholesterol, and so we manufacture it. An adult male, 4 grams a day, 4,000 milligrams a day is manufactured. There's a feedback mechanism so that if you eat eggs and meat are high cholesterol foods. You cut down on the amount manufactured in the liver and continue to have essentially the optimum amounts of cholesterol in the body. Of course, there are people with the genetic disease hypercholesterolemia who manufacture so much cholesterol that it causes a lot of trouble, especially with respect to heart disease. But ordinarily, the body handles that problem of providing the right amount of cholesterol. There are also some people who don't manufacture enough because of genetics, have hypocholesterolemia. Well, that's an example of an orthomolecular substance. And this name became popular among the megavitamin crowd, and pretty soon there was an American Society of Orthomolecular Physicians and the Journal of Orthomolecular Medicine. Well, it started first was the Journal of Orthomolecular Psychiatry. When I first got interested in 1967 in this matter, I thought, how many substances are there that affect the brain and are normally present? I wrote a paper, Orthomolecular Psychiatry, in which I discussed 15 or 20 substances, orthomolecular substances present in the brain, and discussed various aspects from the physical-chemical point of view, various aspects of the question of optimum amounts of these substances, pointing out, for example, that even if we manufacture a substance for ourselves, in general, we don't manufacture the optimum amount because to reach the optimum amount, we have to put in more energy and materials, and if the added gain is not very great, it's better not to strive so hard to get exactly to the optimum amount. Consequently, even if you make substances, important substances, to have a supplement of the important substances is valuable. Well, I'd better get around to cardiovascular disease. In my books, for example, How to Live Longer and Feel Better, I mention an involvement of ascorbate with cardiovascular disease. Emil Ginter in Czechoslovakia pointed out that one important hydroxylation reaction that uses up vitamin C is the hydroxylation of cholesterol to bile acids. The liver keeps manufacturing it 4 grams a day, and vitamin C is involved in getting rid of it by converting it to bile acids, which are then eliminated. And the level of cholesterol in the blood is determined by the rate at which it's being eliminated. That's, you fill up the barrel. If you cut down the rate of outflow, then the level in the barrel goes up higher and you have more cholesterol. So vitamin C, as Skinner pointed out, in extensive studies that he carried out with patients with high cholesterol, high intake of vitamin C lowers the cholesterol level, presumably mainly by that mechanism of speeding up the rate of conversion to bile acids. I put in 2 or 3 other arguments about vitamin C having value against heart disease, perhaps some importance. Then something really important happened, and I didn't know about it for a year or 2 after it happened. I had known a young man, medical student, for about 10 years. Well, he wasn't doctor originally, Matthias Rath. And from time to time I gave him advice For example, I told him that he owed it to himself to settle down and get his MD degree and so on. And he says that I helped him to do that. And he says that I also suggested that he might be interested in doing medical research. He became a cardiologist, began working on a problem, and he and his associates, when they published the results, he was the principal author, came out 3 years ago with the results of a project that they had worked on for several years. There is a protein, a lipoprotein in the blood that is usually neglected. If you send in blood for analysis, the laboratory will report on HDL and LDL and VLDL and not make any mention of lipoprotein A, small a, LPA, which is present in people in amounts considerably less than the amount of LDL. What Dr. Rath and his associates proved is that LDL itself is not what is laid down in the blood vessels in atherosclerosis. It is this other minor component of the blood, lipoprotein A, that is laid down in the arteries. So this, I thought, when I heard about it, was quite a discovery. And I learned from Dr. Rath that lipoprotein A is a very interesting substance in several respects. First, you can understand why people would think that LDL was laid down, because lipoprotein A is a larger lipoprotein than LDL and is made by hooking on another apoprotein to the apoprotein of LDL. The apoprotein of LDL is called ApoB, and it forms a layer around the lipid globule. You hook on another apoprotein about as big as ApoB, which is called ApoA, apoprotein A, small a. And then you have a lipoprotein with twice the surface area, somewhat larger lipoprotein, more lipid in it. And this is what is laid down in atherosclerotic plaques. Well, if you treat an atherosclerotic plaque with antibody against ApoB, you get combination, because there's the ApoB in lipoprotein A. And you can understand that the investigators would have decided that it is LDL that's laid down. But if you treat the plaque with antibodies against ApoA, you also get combination to the same amount. And studies have shown that there's no LDL itself that is laid down in these plaques. It's just the lipoprotein A. There are binding groups on the arterial wall that combine with the apoprotein A and hold it down. Another odd thing about apoprotein A is that it is present, although essentially all animals manufacture it to some extent, it is present in only small amounts in the bodies of most animal species. It is essentially only animals that are deficient in vitamin C, animals that don't manufacture their vitamin C, that manufacture large amounts of apoprotein A, have lipoprotein A in their blood in large amounts. And the conclusion here is that lipoprotein A is present in the blood as a response to ascorbate deficiency. The guinea pig, for example, doesn't manufacture ascorbate. You have to give it vitamin C to keep it alive. And in the laboratories that use guinea pigs are given a diet that contains about 50 times as much vitamin C as would correspond to a human intake, ordinary human intake, because that keeps them in better health than to give them the RDA for humans in their diet. Well, nobody had measured lipoprotein A in guinea pig blood. And Dr. Rath did measure it after we had, in the discussion, had decided that there was good evidence that there was a relationship between vitamin C and apoprotein A. And it was found that, in fact, guinea pigs do have a lot of lipoprotein A in their blood the way that human beings do. Moreover, that you have in the guinea pig a good model that had been barely recognized for atherosclerotic heart disease. Most animals, or some experimental animals, if they are given tremendous amounts of cholesterol in their diet, develop plaques. They have atherosclerosis, but it's not the same as human atherosclerosis, more generally distributed, not concentrated near the heart and coronary arteries. And although it is studied, it's not a good model. Same thing is true of a guinea pig, given the ordinary guinea pig diet that contains a great amount of vitamin C. If you put a guinea pig on a human, in the RDA for humans, then it rapidly develops atherosclerotic plaques in the neighborhood of the heart, the coronary arteries. So it's a good model for the cardiovascular disease for atherosclerosis. The hypoascorbemic guinea pig can be used to study atherosclerosis similar to human atherosclerosis. Well, there is a question, what is the relationship between vitamin C and lipoprotein A? Lipoprotein A, apoprotein A, Dr. Rath and I decided, is a surrogate for vitamin C, a response to the fact that human beings and a few other animals do not synthesize ascorbate and suffer from hypoascorbemia. One thing, if you are on an ordinary human diet, you don't make enough collagen to keep the blood vessels really strong. And in regions of particular stress, such as close to the heart, where the pressure is caused by contraction, the heart is greatest, you get ruptures in the wall. I think cardiologists accept that the primary step in atherosclerosis is a tear in the arterial wall. And then a whole sequence of events takes place. The body tries to control this, to mend this lesion. Fibrinogen are laid down. There are many things that happen. And lipoprotein A is laid down. We think that the principal function of lipoprotein A that caused it to be manufactured in large amounts in human beings and their predecessors and related species is to strengthen the arterial wall. The amount of lipoprotein A in the bodies of different people varies in a remarkable way. With most substances, cholesterol, for example, although human beings differ somewhat in the amount of cholesterol they manufacture, the range isn't very great except for those mutants with hypercholesterolemia who manufacture far too much, and then a few people who manufacture too small an amount. But with lipoprotein A, this sort of control of the rate of synthesis has not yet been achieved. So that if you study population of human beings, you find a considerable fraction with rather low, almost zero lipoprotein A, then a fraction with a medium amount, then a fraction with much larger amounts. Epidemiological studies have shown that first, that lipoprotein A is the strongest risk factor for cardiovascular disease. LDL LDL, cholesterol levels. Cholesterol is a risk factor, all right, but not so strong a risk factor as lipoprotein A. There are some people who don't make very much lipoprotein A, less than 10 milligrams per deciliter in the blood. And they are apt to suffer from heart disease because, I would say, they do not make enough to keep the arterial wall strong. And they're not serving well enough as a surrogate for the deficiency, as a surrogate for ascorbic acid, to rectify the failure to manufacture enough collagen. So those people have cardiovascular disease. Then there are people who make between 10 and 20 milligrams of lipoprotein A in their blood. They have the lowest incidence of cardiovascular disease. And then there are the people who have overshot the mark by manufacturing more than 20 milligrams per deciliter of lipoprotein A per deciliter. And they've been too eager to control the ascorbate deficiency and lay down too much lipoprotein A, occluding the lumen of the blood vessels and leading to coronary heart disease, and in the brain to attacks of stroke. So the primary cause of cardiovascular disease is ascorbate deficiency that leads to weakness of the arterial walls because of the failure to manufacture enough collagen. Then a whole series of reactions takes place, laying down fibrinogen and laying down lipoprotein A and bringing in various other physiological factors. In the case of people with too much lipoprotein A, the atherosclerosis sets in, and often with fatal consequences. Vitamin C, of course, was involved in the development of the ability to manufacture a large amount of lipoprotein A, apoprotein A by human beings and a few other animal species. It's also involved in another way. If a person increases the intake of vitamin C, the level of lipoprotein A in the blood goes down. You no longer need to manufacture so much lipoprotein A, and there's some mechanism that causes the rate of synthesis to decrease, so that vitamin C enters also indirectly in the control of cardiovascular disease. there's a third way another way of handling the problem of atherosclerosis and cardiovascular disease. It is known that the haptenic groups on the wall of the arteries with which apoprotein A combines are lysyl groups. And of course, then if you had the lysine molecules present in the blood, they could combine, and they're present in the blood of every person because lysine is an essential amino acid that you get in your foods, especially meats, fish. But if you have lysine in the blood, these molecules could combine with the combining groups of the lipoprotein A and keep the lipoprotein A from being deposited in atherosclerotic plaques, and even operate to remove lipoprotein A from the plaques to cause the dissolution of the atherosclerotic plaques. So lysine itself, one of the essential amino acids, is a prophylactic measure for preventing atherosclerosis and consequential and related diseases. I had an interesting experience of being approached in April by a biochemist, a leading American biochemist, saying, could I give him some advice? He said he's been taking 6 grams of vitamin C a day, but it doesn't do the job for him that he hoped something would do. He went on to say his brother and his father died young of cardiovascular disease. He was retired early from his job. He received the Medal for Science, the National Medal of Science, in recognition of his work in biochemistry, but he was retired, and he has had 3 bypass operations. His physician has told him that he can't have any more. The saphenous veins have been used up and his health is poor. He likes to walk for exercise. He could take walks by taking a nitroglycerin tablet when he started, and after a while, when angina set in, take another nitroglycerin tablet. Was there anything more he could do? So I said, well, why don't you take some L-lysine, the amino acid L-lysine? He said, how much? I didn't know any clinical results that would help me. One thing I knew was that William C. Rose in the 1930s, when he studied the essential amino acids, had shown that for healthy young men, 800 milligrams of L-lysine in the food, 800 milligrams a day is enough to preserve nitrogen balance. So I thought, well, 800 milligrams, I doubt that it's toxic. I don't remember ever having read anything about toxicity. I'll suggest that you take 5 grams, 5,000 milligrams a day of L-lysine. Start with 1,000 and build up to 5,000. So I got a letter from him saying it's really miraculous. A month after I'd reached 5,000 milligrams a day, I could walk 2 miles without any angina. A month or 2 later, he telephoned me and then wrote me saying that he was feeling so good that he had cut down a tree and was chopping it up for wood and developed angina. So he went up to 6 grams of lysine per day, and he was able to continue rather vigorous work with 6 grams, not being bothered anymore by angina. So I still hear from him. He's a biochemist. He's become quite interested in lipoprotein A, and which he hadn't known anything about. It was discovered in by the way, what time is this now?
That's 20 after 9. 9:20.
How much?
9:20.
9:20.
9:20. 20 minutes after 9 o'clock.
Okay. It was discovered in 1964 by a Norwegian investigator, Cora Berg, and it's attracted a great amount of attention. There are 1,000 papers published, perhaps. Nothing like the literature on vitamin C, but still pretty extensive literature. A lot is known about it. But it remains for Dr. Rath and his associates to discover how really important it is, that it is the cause of atherosclerosis in the people who have too much. It's essentially only the people with more than 20 milligrams per deciliter of lipoprotein A in their blood who are at risk for cardiovascular disease. Those in the second quartile, 10 to 20 milligrams, are protected almost completely, and the ones don't have enough lipoprotein A have a significant incidence, but not only a fifth, perhaps, of the risk of those that have too much lipoprotein A. So we recommend that people who have high lipoprotein A or who have a family history of cardiovascular disease take not only large amounts of vitamin C, which of course I recommend to every human being, at least 2 or 3 grams a day for an adult, proportional to body weight for children, but also take regular prophylactic amounts, perhaps 2 grams a day of lysine, the rest of their lives, of course. That's, don't take vitamin C in large amounts for a while and then stop taking it and go back to poor health again. Well, I'll show my slides. Go through them quickly, and perhaps there'll be time to answer some questions.
There's a keyboard on the podium. It's a touch keyboard.
Anyone have a question while we're waiting for slides? Any questions?
Dr. Pauly,
Yes.
does vitamin C increase the incidence of kidney stones?
No, no evidence it does at all. The vitamin C is the ascorbate ion, and you can't take vitamin C by itself because of its negative electric charge. You have to have a counter ion. And this can be hydrogen ion or sodium ion or something. Well, hydrogen ion, increased concentration of hydrogen ion increases the chance of having kidney stones of less common kind, cysteine stones. Sodium ion acts in the other way to raise the pH of the urine, and it increases the chance of forming the most common stones. Perhaps the best thing to do, it's these counter ions that have some involvement with kidney stone formation, not ascorbate, which helps prevent kidney stone formation. There's no case known of a person having developed kidney stones because of high intake of vitamin C. That's another one of these false statements that you continue to read, such as if you take more than 150 milligrams of vitamin C a day, you're just wasting your money because you immediately eliminate all of the urine. The first experiments that I carried out, which were on myself, I wasn't accustomed to doing experimental work for 50 years perhaps, because I had a lot of students and postdocs, and it was a waste of my time to do it. But I checked myself. When I was taking 10 grams of vitamin C per day, I was eliminating 1.5 grams in the urine. That wasn't wasted, it was helping to protect the urethral tract, of course, against infections. Well, here's LDL. Has a protein, ApoB, that is its outer layer and has lipid. And like protein A, has another protein attached, shown at the bottom part. That doubles the area of the glycoproteins. Next slide. ApoA is much like plasminogen. It has a whole sequence of groups that appear in plasminogen. Lipoprotein is a surrogate for ascorbate. The main evidence is that it's animals that are deficient in ascorbate who produce large amounts of LPA. At the genetic level, I think when the primates lost the ability to make ascorbate, mutants who increased their synthesis of ApoA began to win out, and so the plasma LPA went up in these mutants in man and other primates. With most mammals, they manufacture a lot of ascorbate and don't bother to manufacture apoprotein A. Next slide. At the metabolic level, ascorbate works too to help control LPA. With low ascorbate, you have high plasma LPA. In cancer and other serious diseases, the ascorbate level drops because the doctors don't know enough to give the patients large amounts of vitamin C, and it's used up in the fighting of the disease, and the plasma LPA goes up. Diabetes, same thing. And if you increase plasma ascorbate, the level goes down again. Next slide. Well, ascorbate deficiency. If you're low on ascorbate, you don't synthesize enough collagen. You get those ruptures that are the first manifestation of imminent atherosclerosis, and then various changes occur in the walls. Next slide. Here, 10 years or so, about 10 years ago, Emil Ginter published essentially this diagram. There had been a decrease in death rate from cardiovascular disease, coronary heart disease in the United States. And at the same time, over the same period, a great increase in the intake of ascorbate by the American people. I think that's the last slide. So, any other questions? Yes?
I noticed that in formation of LPA there is disulfide bridge between the ApoB and the plasminogen-like other part. Could it be that ascorbate regulates the formation of LPA by being the major reductant in plasma?
Well, probably the ascorbate is the principal reducing agent in the human body, and there's no doubt that changing the ascorbate dehydroascorbate ratio by taking large amounts of vitamin C changes the oxidation reduction levels in the body in such a way that there'd be a greater tendency to convert the disulfide to two hydrosulfide groups to split this bond. Yes, I think so. There hasn't been a large amount of work published on this point, but I think that it occurs, and it may be that one of the ways that I haven't discussed for ascorbate to control cardiovascular disease is its function as an antioxidant in preventing the peroxidation of cholesterol in the plaques. It's the peroxidized cholesterol that is especially damaging, and ascorbate can function directly to prevent this peroxidation, and indirectly there are many disulfide bonds in the ApoA molecule, and I'm sure that often they are split open to hydrosulfides, which are right there in the lipoprotein A and adjacent to the cholesterol and help prevent peroxidation. Another.
Would a high intake of ascorbic acid lower serum cholesterol?
But
Would a high intake of vitamin C lower cholesterol in the serum?
Lower serum cholesterol.
well, is by speeding up the rate of conversion of cholesterol to the bile acids. And there are perhaps other ways too, but I don't know enough to be able to say. Do you have anything to say, Dr. Rath, on that point?
Well, there are a series of studies where it has been shown that vitamin C, in fact, it occurs over a prolonged period of time, so that the body metabolism has to adapt to that.
Any other questions? Yes.
How accurate is the average length of the measurement?
Well, how accurate do you think the measurement is?
Well, there are a few commercial kits out now, basically based on an ELISA or radioimmunoassay. The problem with these assays is that they measure only the ApoA protein. And what has not been discussed because of the time constraint is that ApoA is like a chameleon. It varies between individuals in size dramatically. So there are about 12 isoforms known, and probably there are 50 isoforms varying between individuals. So some of them are extremely short, some of them are 5 or 10 times the size. What I'm aiming at is that probably the future belongs to an ELISA system where the ApoA is bound and the ApoB, which is the other part of the molecule, is detected with a second antibody that is coupled to peroxidase or anything.
There is a point that I failed to mention when I was talking about lysine as an inhibitor to the binding of lipoprotein A to the vascular wall, to lysyl groups. Lysine is not a very good inhibitor compared with other antifibrinolytic agents, drugs such as tranexamic acid, for example, or 6-aminohexanoic acid, that's epsilon aminocaproic acid. These are much stronger inhibitors, and so far as I'm aware, no one has used these drugs together with vitamin C in the treatment of serious atherosclerosis. We think that they should be used, that this is could be an effective treatment, more effective than just giving lysine. But of course, it's lysine that we recommend for prophylaxis, that persons who are at risk for cardiovascular disease should take lysine regularly every day. Well, one more question. You can get additional information. We have 4 papers in the last issue of the Journal of Orthomolecular Medicine. I understand that the medical school library has a copy of this issue. I don't think they subscribe to the journal regularly, but they have a copy of that issue. Well, we have copies of these papers that anyone can get by just calling our institute and asking for them.
There are about 20 copies which we posted on this chair, so anybody who's interested right now.
Yes, sir. You can pick up one of these pamphlets if you want. It has the article about the biochemist who controls this engine with lysine.
Dr. Pauling, thank you very, very much for
I think that Dr. Rath and his associates made a great discovery, and that it's going to lead ultimately to really significant control of cardiovascular disease. Well, I have